Hello everybody, my name is Patrik and with my colleagues we prepared for you this presentation about our first step towards a DNA barcode reference database for mayflies of Slovakia. So, although mayflies represent a relatively small order of amphibiotic insects, their larvae provide several essential roles in freshwater habitats. First, as periphyton and detritus consumers, an integral part of the diet for fish and other aquatic invertebrates, they serve as link in energy flow between primary producers and secondary consumers. In this way, they contribute to nutrient cycling and spiraling in freshwater ecosystems. In a few exceptional cases, as shredders, they also provide the decomposition of coarse particulate organic matter. By reworking sediments in rivers and flushing waters through their caves, the burrowing species contribute to bioturbation and bioirrigation. Representatives of filter feeders mayflies contribute to freshwater ecosystems' self-cleaning processes. Finally, due to their cosmopolitan distribution and high-quality water requirements, they are also important ecosystem health indicators and integral part of the biomonitoring protocols. Thanks to the great initiative of well-known ephemeroptologists such as Professor Landa, Dr. Soldan and Dr. Devan, the Slovak mayfly fauna is relatively well researched. According to the latest known data, there are 123 species from 33 genera and 16 families in Slovakia. However, re-evaluating dubious findings and incorporating current knowledge into the taxonomy could reduce the number of valid species. It is also necessary to verify the occurrence of another 10 species that have not been recorded for more than 3 decades. Compared with faunistic and ecological research of mayflies in Slovakia, genetic studies have not received much attention. Even though the BOL database contains more than 38,000 records of mayflies from 56 countries, data from Slovakia, mostly situated in the geographically attractive region of Western Carpathians, are still lacking. Simultaneously, the territory belongs to the ecoregion of Carpathicum and Pannonicum defined by Ilias. Due to these two facts, the biota of this area is remarkably diverse and enriched by the overlapping influences of alpine, Arctic alpine, Mediterranean, and Asiatic regions and serve as an important center of endemism. Accordingly, we began to focus intensively on the DNA barcoding of mayflies from Slovakia approximately 2 years ago. In the first step, our research required numerous fieldworks, and to date we have managed to sample 142 different sites. The collections took place in a wide range of habitats from karst springs to the potamal sections of the largest Slovak rivers and from the lowlands to the alpine lakes in the Tatra Mountains. Until now, we were able to process individuals from 47 sites marked with a green label on the map. Primarily, we determined 42 species from 10 families using morphology, with the most species recorded in Heptagenidae and Betidae. DNA was extracted from at least 3 individuals per site per species to capture also intraspecific genetic diversity. Finally, DNA was extracted from 502 individuals, and the target CO1 barcoding fragment was obtained from 403 individuals and uploaded to the BOLT database. The sequences are included in the BOLT projects Ephemeroptera of Slovakia 1 and 2 with these two codes. In total, 403 sequences of 42 species were finally assigned to 62 bins, whereby sequences of 12 morphospecies were divided into more than one bin, indicating the presence of cryptic lineages or species. The sequences of 23 individuals belonging to 5 species created 7 unique bins and currently represent the unique genetic information for Slovakia. 2 unique bins were created by representatives of Habroleptoides confusa and Betis rodani. At the same time, these bins are the nearest neighbors to each other. In the first mentioned species, the barcoding gap is 1.73% and more than 2% in the case of Bettis rhodani. The beans created by the representatives of Canis bescidensis and Bettis buceratus represented a much bigger gap. However, beans with only one sequence must be verified by further records. Here, on the left side of the slide, you can see a maximum likelihood cladogram of 62 beans with the highlight rectangles A to L, which represent species assigned to more than one bean. The asterisks indicate already mentioned unique beans. However, CO1 marker is not suitable for analyzing family-level relationships and therefore are some families paraphyletic, such as Leptophlebida and Ephemeralidae. The slide's right side displays the 12 species from 5 families representing the species with high intraspecific variability or cryptic lineages, which created more than one bean in the bulk database. The highest number of beans were created by widely distributed species such as Bettis rhodani, 6 beans, Habroleptoides confusa, 4 beans, and Ecdionurus venosus, 3 beans. Percentage values for individual species correspond to the maximum intraspecific variability of Slovak specimens calculated by the Kimura-2 parameter. Some representative resulting values were surprisingly high and far exceed the CO1 level of intraspecific variability based on the both standards. For Ecdionurus venosus, Canis luctuosa, and Bettis multicus, these values well exceed 20%, and for Ecdionurus torrentis, Bettis rhodani, and Bettis vernus, it was about 10%. On this slide, we look closer at the population structure of some representative of genus Ecdionurus. We can notice that 3 phylogenetic lineages or beans of Ecdionurus venosus and 2 phylogenetic lineages or beans of Ecdionurus torrentis do not form the monophyletic clusters on the phylogenetic tree. However, the bootstrap values of some nodes are relatively low. Therefore, we create a haplotype network to verify the relationship between the 7 phylogenetic lineages of 4 species from the genus Ecdionurus. In the haplotype network, we can see that 3 lineages formed by 13 sequences of Ecdionurus venosus represent one haplogroup separated by 67 mutations from Ecdionurus torrentis. However, 6 sequences of Ecdionurus torrentis did not confirm this assumption. 3 haplotypes created by the lineage SK1 were closer to the species Ecdionurus maccani than the quite distant haplotypes of lineage SK2. However, if we look at the species photograph representing 7 lineages, it is possible to see that several individuals were identified only using larvae of a relatively early insert. This fact is especially evident from their relatively short wing pads, such as on the photo of Ecdionurus torrentis SK1 and SK2. Therefore, it is possible that these individuals were also incorrectly determined using morphology. Nevertheless, hybridizations cannot be excluded as well. We also took a closer look at the population structure of 6 phylogenetic lineages of Bettis rhodani. 54 individuals finally formed 29 haplotypes, whereby lineage SK5 included 20 of them and represent the most abundant lineage in Slovakia and Europe. At the same time, this lineage is separate from the closest haplotype up to 81 mutations. 14 mutations separate haplotypes of 2 unique beans from each other. As we can see on the map, these haplotypes were recorded from 5 sites situated in the northwest part of Slovakia. On the 2 localities, we confirmed their coexistence. We observed a similar case in the 3 other localities where we recorded the coexistence of the lineage SK4 and SK5, SK3 and SK5, and SK2 and SK5. To sum up, our study indicates evident importance of more detailed sampling and DNA barcoding due to some mayflies' unexpected intraspecific genetic diversity representing several beans some of which may be rarer than others. Therefore, their records in the DNA reference libraries are necessary because they might be evaluated as sequencing errors during the bioinformatic processes in metabarcoding or eDNA analysis. Thus, we can lose a unique part of genetic diversity. Concurrently, we confirmed that Slovak samples barcoding contributes new data to the international databases, and it also makes sense to barcode species that are already barcoded. Mayflies, as well as other groups, confirm the need for more intensive barcoding in the Carpathian Pannonian region because of their unexpected hidden freshwater biodiversity. Finally, this contribution was supported by these two grants, and concurrently, we would like to thank our colleagues from the Zoo Lab of Plant Science and Biodiversity Center of Slovak Academy of Sciences and the Department of Ecology at the Faculty of Natural Sciences of Comenius University of Bratislava because of their always helpful and pleasant atmosphere during the work. Many thanks also goes to the team of Professor Michal Grabowski from the Faculty of Biology and Environmental Protection at the University of Lodz, who enabled me to molecularly process most of the samples during the short-term scientific studies carried out through the COST project. At the same time, thank you for your attention and we wish you a pleasant rest of the conference.